Okay, we're moving on to a new chapter 8, cellular reproduction, cells from cells. So this will be 106 cell repro 1. And so we're going to find out why cellular reproduction matters. Why do we need new cells? Which cells undergo this process, which cells don't? And we're going to look into asexual and sexual reproduction. And there's going to be two types of cell reproduction, and that sexually reproducing organisms do both of them. One's called mitosis, which is an asexual process. And if they're a sexual reproducing organism, which most organisms are, they also have a second cell reproduction specialized one called meiosis. Okay, and so they're going to go in the start off with asexual reproduction. So zookeepers at the Chester Zoo were surprised that that Komodo dragon had laid eggs. And there was a female Komodo dragon, and she had not had relations with a male, and yet she's laying eggs. This is called parthenogenesis, or virgin birth. This is a process of making eggs by a process called mitosis, which is an asexual process. And this is what normally somatic cells do. Somatic cells are the cells in your body that are involved with reproducing of their individual. And usually in sexual reproduction, you have to make specialized cells called gametes in a process called meiosis. And that's what a Komodo dragon would usually do. But some organisms that, even though they do sexual reproduction, they occasionally can go into an asexual means. And this is parthenogenesis. Production of offspring by a female without involvement of male. And DNA analysis confirmed that Flora's offspring derived their genes solely from her. In sexual reproduction, half your genes come from mom, half your genes come from dad. In asexual, they all come from mom. So in some organisms, that's all they do. They just do parthenogenesis. And they only need one parent, mom. And so the entire population there's some lizards like this and like dandelions. There aren't any male dandelions. They're all female. The entire population is female. Now this, of course, Komodo dragons usually reproduce sexually. So of course there are male Komodos. But every once in a while, the female can produce eggs by this asexual process, and her offspring are related to her 100% because they have the same genes she has. There were no genes from dad. Life with or without sex. We're going to find out that sexual reproduction is by far the more popular means of reproduction, and for reasons that actually are not that clear as to why it's so popular, because it has huge disadvantages compared to asexual reproduction. We're going to get into that later on. Okay, so documented in sharks, domesticated birds, and now Komodo dragons. Never documented in human beings, though. And so this is parthenogenesis among organisms that usually reproduce sexually. Some have gone completely to parthenogenesis, but that's actually pretty rare. Like I said, there's a certain type of lizard in the American Southwest that's parthenogenic only. And then dandelions in your lawn, they're completely parthenogenic. They've done away with sex. Okay, so remember, this is a characteristic of life, procreate, to make new individuals of the same species that distinguish living things from non-living things. All organisms, from bacteria to lizards to you, are the result of repeated cell divisions, making new cells. Remember, you started off life as a single cell called a zygote. And where are you now? You're hundreds of trillions of cells, and they all come about by this process called mitosis, which is asexual cell reproduction. So the perpetuation of life therefore depends on cell division and the production of new cells. Because remember, that was part of the cell theory we got back in the cell chapter, the introduction to the cells, that right now all cells come from pre-existing cells. Not going to be new cells arising from non-living stuff anymore. Remember, not in an oxygen environment. Okay. Reproduction may result in the birth of new organisms, but more commonly involves the production of new cells. And we're going to see, you know, why do we need new cells? Of course, when we're growing up, from a baby to a toddler to an adolescent, we're making new cells and we're growing. Because when you're an adult, you stop growing this way, right? You may still grow this way, but you stop growing this way. But you're still making new cells. Some of your cells, we're going to learn that some of your cells in your body still undergo mitosis to make new cells. Other cells have become amitotic. They've stopped mitosis. They're not going to do it anymore. And so those are the cells you don't want to lose, because if you lose those, you don't get them back. And this includes most nerve cells and skeletal muscle cells and cardiac muscle cells. Smooth muscle cells you make more of, but not skeletal or cardiac muscle. This is part of what I talked about in the at-home mitosis lab. Okay, when a cell undergoes reproduction or cell division, two daughter cells are produced that are genetically identical to each other. Identical to each other and the mother cell, the parent cell, genetically identical. This is what makes it an asexual process, because the resulting cells are identical. In a sexual process, the resulting cells are not identical. Okay, before a parent cell splits in two, it duplicates its chromosomes. It's like I said, so a cell, let's use a human cell, for example. I'll always use a human cell, has 46 chromosomes, right? That's the parent cell. Okay, so it divides in two. How many cells, how many chromosomes do the daughter cells have? 23? Does it split in half? Well, then what about those cells? When they go, what do they split? What do they end up with? 11 and a half chromosomes, and then 5 3/4 chromosomes, and so on and so forth? No, the resulting daughter cells have this exactly, remember, they're identical to the parent cell and identical to each other. So they have exactly the same number of chromosomes. So if a human cell is going to go through mitosis and divide in two and to make two cells, it has to replicate its chromosomes. It has to really make 92 chromosomes. So each daughter cell has 46, right? So, and this is called, this happens in what's called the S phase of the cell cycle. So during cell division, each daughter cell receives one identical set of chromosomes from the lone original parent cell. It's the same number. Okay. So cell division plays important roles in the lives of organisms. What is it? Replaces damaged or lost cells. For some things, if you lose some skin cells, some connective tissue cells, you make some new ones. You lose some blood cells, you make some new ones. But as I said before, when you're an adult, there are certain cells that you can't replace, like nerve cells, most nerve cells. So if you lose them for good. Permits growth. As I mentioned, you start off as a little tiny baby, you went to a toddler, adolescent, you grew. And also as a zygote to an embryo to a fetus, you added lots of cells, you grew. And then allows for reproduction of the whole organism as a whole. If you're asexually reproducing, you use that same process, mitosis, to make babies. If you're sexually reproducing an organism, you have to use another process called meiosis. That's a different and specialized process. And we're going to get to that after we discuss mitosis. Then we're going to discuss meiosis and the differences in where that happens and when it happens. Okay, functions of cell division by mitosis. Human kidney cell, you lose a kidney cell, you can replace it. Early human embryo, like 8-cell embryo to 16 to 32 and so on and so forth. Reproduction now, and then, so this is cell replacement and growth. Right? And down here is asexual reproduction for those organisms that conduct asexual reproduction. Now, there are some organisms, usually primitive ones, that can do both. They use asexual reproduction for the most part, but then they have a sexual side to them. So they can do both. Humans are obligate sexual beings. We can't make babies any other way but by sex. Okay, so the amoeba right here divides in two by mitosis. That's asexual reproduction. Here, regeneration of a sea star. You cut off one of the arms of the sea star. That sea star regenerates that arm, but then the arm regenerates the rest of the sea star. So this is called fragmentation. And this is a form of asexual reproduction. Same thing with growth of a clipping is almost identical to that, because you can take some plants, you break off a branch, you stick that branch in the ground, that branch grows roots, and that branch becomes a whole other organism. So this is really fragmentation in plants, rather than fragmentation it's really a form of fragmentation, growth of a clipping. Very similar to regeneration of a sea star. Okay, in asexual reproduction, single cell organisms reproduce by dividing in half. And there's no fertilization of an egg and sperm. We don't talk about eggs and sperm, we talk about asexual reproduction. And as you just mentioned, many multicellular organisms can reproduce asexually and sexually. So the sea star that was able to grow a new organism by the severed arm growing a new sea star, they have male and female sea stars, and they can have sex. They have gonads, they make eggs and sperm, and they often have sex, but they can fragment and use asexual reproduction. The same thing with a plant. The plant is a sexual organism, but if you break off part of it and plug it in the ground, you can make a new individual by asexual means. Okay, in asexual reproduction, one parent, and its offspring, it makes offspring asexual, which means the offspring are identical, genetically identical in every way. There's very little variation. Now, there is variation by mutation. Now, we're going to get into that later on, about how when you copy DNA, you make mistakes. There's mutations. So all organisms change over time. This is evolution. In that sense, evolution is a fact. Organisms change over time because of mutations, about these errors. So even asexually reproducing organisms change over time. But sexually reproducing organisms change faster. Okay. Oh, I get the hiccups. No. Mitosis, the type of cell division responsible for asexual reproduction and growth and maintenance of multicellular organisms. Sexual reproduction requires fertilization of an egg by a sperm, and these are called gametes. Involve a special type of cell division called meiosis, which only occurs in reproductive organs. So in your ovaries, if you're female, your testes, if you're male, this is the only place where this sexual process, this sexual cell division is taking place. And they make gametes that have half the number of chromosomes. So in this case, you start off with a cell with 46 chromosomes. You do make cells with 23. And these are called gametes. And they're called haploid because they have half the number of chromosomes that a diploid cell would have. So in human beings, that would be 46 is the diploid number, 23 a haploid number. If you're an onion, 16 will be the diploid number, 8 will be the haploid number. Okay, so in summary, two types of cell division are involved, in the lives of sexually reproducing organisms. One, mitosis for growth and maintenance, and two, meiosis for reproduction of a new individual. Okay, so in the eukaryotic cell, most genes are located on chromosomes in the cell nucleus. Okay? A few genes are found in DNA in mitochondria, and in those autotrophs, eukaryotic autotrophs have a chloroplast, and they have some genes in those also. Remember, mitochondria and chloroplasts actually have a chromosome in them. So each eukaryotic chromosome contains one very long DNA molecule, typically bearing thousands of genes in a longitudinal arrangement, one after another. The number of chromosomes in a eukaryotic cell depends on the species. It's just saying that our number is 46. If you were a chimpanzee, it would be 48. If you're an onion, it would be 8. I think if you're a fruit fly, it's 4. It's always even. Why is it always even? Because you always get the same, in sexually reproducing organisms, they always get the same number of chromosomes from dad as from mom. So you put those together, it's always an even number. And here the number, well, here I was giving you a summary here. So as I said, human 46, mouse 40, giraffe 30, possum 22, is bison 60, dog 78. Just because you have more chromosomes, though, doesn't mean you have more genes, because there are huge spaces in chromosomes where there aren't any genes, and so just because you have actually your diploid number is higher doesn't mean you have more genes. Because look, this red biscacha rat has 102 chromosomes. That's probably because it might be triploid or tetraploid. It may have 4 sets of chromosomes. Usually we are diploid, we have 2 sets. But there's some plants that have like 3 and 4 sets, and there's very few animals. There's a couple of animals that have 3 or 4 sets. So they just have more copies. They don't have more genes, they just have more copies of the genes. I think we knew this already. Chromosomes are made from chromatin. In eukaryotes, fibers composed of roughly equal amounts of DNA and protein molecules, which helps to organize the chromatin and control the activity of its genes. Most of the time, the chromosomes exist as thin fibers that are much longer than the nucleus they are stored in. And you can't see them under a light microscope. So one of these DNA, one of these cells could be 6 feet long, but it's really crushed into a tiny little space. As a cell prepares to divide, its chromatin fibers coil up, forming compact chromosomes that can be viewed under a light microscope. And if we still do in a microscope lab on campus, we would actually look at a microscope and see these chromosomes. When a cell is ready to make new cells in mitosis, it really supercoils and condenses the chromosomes so you can see them. The cell is not dividing. It's in what's called interphase. Chromosomes are too thin to be seen under a light microscope. You can see them with an electron microscope, but not a light microscope. Well, there they are right there. There is probably an onion root tip, which we would see in lab. And see the cells on the sides here, they're not in mitosis because you can't see the chromosomes. This cell is in mitosis, and that's actually what's called prophase. We're going to get to the phases of mitosis in a moment. So when you see those ropey-like things inside a cell, you know it's undergoing mitosis. Okay, so remember, histones are proteins used to package DNA in eukaryotes, and nucleosomes consist of DNA wrapped around these histone molecules. So they are. There's a DNA double helix. Here's the histones, and you have like 8 of them, and you got 2 wraps of the chromosome around these 8 histones in like beads on a string, and that thing's called a nucleosome. Each group of the 8 histones and the 2 turns of the DNA is a nucleosome. And you can, like I said, that's a electron micrograph there. So you can see them on electron with an electron microscope. It is the beads in a string. And there they are supercoiled. So this is what's called a duplicated chromosome. It's right after the phase of the cell cycle when chromosomes are replicated or copied and they become supercoiled. Okay, before a cell begins the division process, a DNA molecule of each chromosome is copied to the process of DNA replication. And new histone protein molecules attach as needed. The result is that each chromosome consists of 2 copies. So you start off with a single chromosome, and they end up with a replicated chromosome. And these two copies are called sister chromatids. They're held together by something called a centromere. So it looks like an H. 2 sister chromatids held together by a centromere, and those sister chromatids are genetically identical. And they came from one that split and made 2. And you have to do that before mitosis can begin. So when a cell divides, the sister chromatids of duplicated chromosomes separate from each other. So you have these sister chromatids attached to each other. And in one phase in mitosis, they are split apart. And they become individual, full-fledged single chromosomes in their own right. So one of the chromatids goes into one cell, it becomes a single chromosome. The other chromatid goes into another cell. So there's chromosome duplication. One chromosome here is duplicated is made into a replicated chromosome. Now, it's important to think, and when we keep things straight, particularly when we look into what's happening in meiosis, we got to make sure that when we count chromosomes, this single chromosome is one chromosome. This replicated chromosome, the 2 sister chromatids, is also one chromosome. It's one replicated chromosome. So you have one single chromosome and one replicated chromosome. So it's important not to think of it as 2 chromosomes yet. Just think of it as one. Otherwise, you get the numbers screwed up when we figure out what's going on in meiosis. So eventually, those sister chromatids will be separated and become full-fledged chromosomes. And then you could say, oh, now we have 2 chromosomes. The cell cycle is the ordered sequence of events that extend from the time a cell is first formed from a dividing parent cell to its own division into 2 cells. So think of the cell cycle as the lifetime of a cell. How the cycle of life for a cell. It's called the cell cycle. And mitosis takes place in one phase of that cycle. And then the rest of the time, it's not going to mitosis, and that's called interphase. And that's divided up into 3 phases itself. So let's look at the cell cycle. There it is, there's the cell cycle. So let's look at a cell that's just going through mitosis. Here we have a cell with single chromosomes, and we go through a phase called G1. And we can think of G as like growth, right? It's growing. And at one point, it comes to the decision, I don't know how it does this, that I want to undergo mitosis. I want to divide into 2, make 2 cells. So then I have to go through DNA synthesis. So when we go from G sub 1 to the S phase, S stands for synthesis of what? DNA. So then we make single chromosomes into replicated chromosomes. Then we go through a phase called G sub 2. And you can think of this as growth also, or it's more like gap, where it's getting ready to go through mitosis. It has to get ready. And when it's finally ready, then it enters the mitotic phase, where it actually becomes 2 cells. So So you have the G sub 1, S, and G sub 2, and collectively they're known as interphase. And this is when a cell is not dividing. So 90% of the time, they're not dividing, and they're in either G sub 1, S, or G sub 2. But of course, remember, some cells, like in adult nerve cells, they're amitotic. They're stuck in G1, and then they never get out of G1. They've turned off mitosis. But if it's a skin cell, for example, you're constantly making new skin cells, and so they're constantly going through the cell cycle and constantly going through mitosis. But 90% of the time, they're in interphase. Okay. So, as I said, most of the cell cycle spent in interphase, lasts for about 90% of the cell cycle for the cells that go through this cycle. Now, sometimes they stay in interphase, and they can go to mitosis if something happens, but they don't. For example, some connective tissue cells. So you have some connective tissue cells in your hand, for example. Well, what happens is you cut your hand, you injure your hand. Well, this actually stimulates mitosis in those cells. So then they go through S and G sub O and mitosis and make new cells to repair that damage. Right? And then they stop mitosis until they get injured again. You see? So some cells in the body are constantly going through mitosis all the time, like skin cells, like the cells lining your GI tract, or blood cells in your bone marrow, red blood cells, white blood cells, and these blood cells that make platelets are constantly going through mitosis, making those red blood cells really only last 3 months. Actually, red blood cells are not really cells, but we call them cells. Okay, during interphase, the cell performs its normal functions, doubles everything in cytoplasm, and grows in size. From the standpoint of cell reproduction, the most important event of interphase is chromosome duplication, where DNA in the nucleus is precisely doubled, and that's called the S phase. Again, S for DNA synthesis. So interphase periods before and after the S are called, as you mentioned, a G sub 1 and G sub 2. G could stand for gap, or it could also stand for growth, I think. During G sub 1, each chromosome is single, cell performs normal functions. During G sub 2, each chromosome in the cell consists of 2 identical sister chromatids, and the cell prepares to divide. Okay, the mitotic process divides 2 overlapping processes. And the book divides this up, and I don't usually do this. Mitosis, in which the nucleus and its contents divide evenly into 2 daughter nuclei, and then cytokinesis, in which the cytoplasm, along with organelles, is divided into 2. I usually don't separate out cytokinesis as a separate process. I think of it as a hallmark, or process that occurs in what's called telophase, which is the last phase of mitosis. That's where cytokinesis occurs. But you can think of it in either way. So the combination of mitosis and cytokinesis produces 2 genetically identical daughter cells. Okay, so figure 8.7 illustrates the cell cycle for an animal cell using drawings, descriptions, and micrographs. Micrographs are on the bottom row. The page shows dividing cells from a salamander. Drawing the top row include details that are not visible in micrographs. The text within the figure describes the event occurring in each stage. So, what's going to happen here is we have a cell in interphase right here. Right? Then we have what's called a mitotic spindle form. These are microtubules that are going to attach to chromosomes and move chromosomes. And in prophase, what happens is the spindle fiber forms, the chromosomes condense, and they get into this jumble of chromosomes. The nuclear membrane disintegrates, and then in late prophase, the spindle fibers attach to the centromeres and start yanking and pulling chromosomes. And where they pull them to, so that's prophase. It's just a jumble of chromosomes, just a random jumble of chromosomes. Then what happens is the spindle fibers pull the chromosomes right into the middle of the cell. One line down the middle of the cell. That's called metaphase. And so all the chromosomes are on one line down the middle of the cell, and the green are the spindle fibers. They're attached to the chromosomes. Then then the centromere splits, and the spindle fibers are like stretched elastic bands, and they pull apart the sister chromatids. And then you have two lines of chromosomes. There's two lines of chromosomes. And now these chromosomes are single chromosomes. They're no longer replicate. They used to be sister chromatids, and now they're single chromosomes in their own right. And then finally, telophase. This is where the chromosomes are pulled to the two poles of the cell, and they become tight bundles and distantly apart in the two poles of the cell. And then the nuclear membrane starts to reform around this jumble of chromosomes, and cytokinesis starts. So the chromosomes and their movements depend upon a mitotic spindle, a football-shaped structure of microtubules that guides the separation of the two sets of daughter chromosomes. Cytokinesis usually begins during telophase, divides the cytoplasm, and is different in plant and animal cells. Because this process is identical in plant and animal cells, except for cytokinesis. Animal cell cytokinesis is known as cleavage, and it begins with the appearance of a cleavage furrow in indentation, and you have a pinching off of these two cells. Like that. But in plant cells, they have a cell wall. So when vesicles containing cell wall material collect in the middle of the cell between these two tight bunches of chromosomes, start forming a cell plate of cellulose. And so the cell plate starts to form and goes all the way, eventually hitting the cell wall on either side. And once it completes that cell wall, then mitosis is over. And you have two brand new daughter cells in G sub 1. And there we have a cell plate forming between the two daughter nuclei. And when it gets all the way across, it becomes two brand new cells. And there it is. Okay, I am going to leave it there and get into the cell cycle control system later on in the next video. So this one, remember, is 106 cellrepro1. So I'll catch you in the next video here.